So, again, yeah, the title is a circuit for deviance detection in primary visual cortex. I want to thank Eric and Terry again for the invitation. And I'll jump right into the general construct that I'm studying. So, oh wait, here we go. So, sensory stimuli and visual stimuli in particular are processed in the context in which they appear. So if, for instance, you were to spot an apple in the context of a bunch of other fruit, you might expect to observe one level of response in visual cortices in the brain. Versus if that same stimulus were to appear in a context where you expect apples and you expect that stimulus, what we actually see is a reduction in the evoked visual cortical response to that stimulus. Versus if you were to observe the same stimulus in a context in which you were expecting something totally different, like a bunch of oranges, what we observe in fMRI and EEG studies is actually an augmented response or a larger response in basic sensory areas. And this has been classically studied in the literature with a very basic sensory oddball paradigms, and this has been done for decades now. And this involves the relatively rapid presentation of either auditory or visual or tactile stimuli. And what you do is you present a standard or a repetitive or redundant stimulus over and over and over and over. And then every now and then present a stimulus which deviates in some way, typically either some physical property like a change in pitch or a different orientation in the visual domain. And these deviant stimuli are rare compared to the standard or redundant stimulus. And the brain responses are recorded in humans with EEG at the scalp. And the most consistent and common finding and thing to study in this paradigm is our evoked potentials. So here, this is just a typical averaged scalp potential from EEG, where you see the thin line is the evoked response to the repetitive or the standard stimulus, and the thick line is the evoked response to the kind of unexpected or deviant stimulus. And what you can see is that the evoked cortical response is much larger to the kind of the rare or deviant. And by calculating a difference waveform between these two event-related potentials, the early and most consistently observed large response is termed the mismatch negativity. So in the auditory domain, this tends to peak around 150 to 200 milliseconds after the onset of the stimulus. The mismatch negativity, importantly, and my question during the first talk was kind of motivated by this. It's thought to be pre-attentive in the sense that it's elicited in the absence of a task or directed attention to the stimuli. So oftentimes to elicit the mismatch negativity, the person will be told to pay attention to some movie on the screen, and the mismatch negativity will be generated by the auditory stimuli, or vice versa. And it's an obligatory task. It's not necessarily the same thing as startle. It's not necessarily the same thing as attention. It's thought to reflect an early sensory prediction error in primary sensory cortices. So why this is particularly important in the human in the clinical literature is that the mismatch negativity, or this kind of increased negative going scalp potential to rare deviant stimuli, is significantly lower in individuals with schizophrenia and other psychotic disorders. So this has been reliably replicated in hundreds of reports over the past 3 decades. It's one of the most consistent biomarkers of the disease, and it's also relatively easy to record. Again, it doesn't require a task or attention. And it's thought to index aberrant processing of basic stimuli in the context in which they appear when it's shown to be reduced in a subpopulation, and it can also be thought to reflect reduced prediction error signals in cortical circuits. And it's present so far regardless of the sensory modality and that's being studied. So while the vast majority of studies in mismatch negativity and schizophrenia have been done in the auditory domain, there is a growing literature of visual mismatch negativity, and the effects appear to be just as robust and consistent. But perhaps what's even more important for the in the clinical domain is that it significantly predicts and correlates with deficits in global and day-to-day functioning of individuals, and it correlates very well with cognitive deficits, reading deficits, and so on. So unfortunately, although mismatch negativity by definition is a scalp measurement with EEG, there's not a clear idea of the essential neurobiology of mismatch negativity in the healthy sense, in a healthy individual, as well as exactly why it's aberrant in individuals with disease. And there's also just not necessarily a clear idea of the basic neurobiology of sensory prediction error, or the kind of the neural or the psychological basis of mismatch negativity. So this is the essential motivation of my work, is to see if we can translate this study, or at least particularly this paradigm, to a mouse model, where we can apply some of these newer neuroscience techniques to get a deeper understanding of the essential circuit basis of sensory prediction error. So that is the general motivation of the talk, and this is what I'm going to go through. First, I'm going to describe the basic methods and paradigm which I use to study visual deviance detection or deviance processing in mice. The majority of the data that I'll present come from awake head-fixed mice on a rotating treadmill, and I use 2-photon calcium imaging of visual cortical neurons to measure their activity. I'll show you that deviance detection signals appear to be primarily present in layer 2-3 pyramidal neurons, and actually even perhaps in distinct subsets of pyramidal neurons in layer 2-3 of mouse visual cortex. And further, I will show you how a higher level mouse prefrontal area, the mouse anterior cingulate area, which sends top-down projections to V1, appears to be active during the basic sensory oddball paradigm, how it synchronizes with V1, particularly in theta and alpha bands, and how this potentially modulatory influence is necessary for the generation of deviance detection signals, which we think are the underlying neural basis of the mismatch negativity. And then I'll pivot to my kind of more long-term and big picture goals, which is to test the specific circuit level hypothesis for how augmented neural responses to deviant stimuli are generated in primary visual cortex, involving interactions of this top-down projection and local interneurons in the primary visual cortex. And so I'll show you some data on how V1 interneuron activity, VIPs and somatostatin positive interneurons are differentially active during the oddball paradigm, and how top-down inputs to V1 drive these different interneuron populations, but differentially, specifically when we drive this circuit at theta alpha frequencies. Hopefully make it that far. Okay, so this is the basic paradigm that we use in my lab. So here, this is an awake head-fixed mouse, and we're typically using full field, 100% contrast oriented grading stimuli, moving 2 cycles per second. They're half a second long, so we just go through a whole cycle. The idea is to present the mice with a consistent, repetitive visual stimulus over and over to the point where that stimulus becomes redundant. And then every now and then throw in a rare deviant stimulus, just like as is done in a typical sensory oddball paradigm. And to measure visual cortical activity, we use genetically encoded calcium indicator GCAM6. And this allows us to quantify the visual stimulus evoked activity of V1 neurons during this paradigm. So here, when you see neurons getting brighter in the image, it suggests they're firing a burst of action potentials. So here is some real-time data from this paradigm in primary visual cortex. And I've modified the stimuli on the left here just to make things a little bit more clear. But on the right, these are neurons in V1. And on the left, these are basic oriented visual stimuli that the mouse is seeing. This was collected when I was postdoc. And in this particular study, I used static gratings, but I eventually moved on to moving gratings for a number of different reasons. But here you can see during the presentation of the repetitive orientation, the vast majority of neurons are not changing fluorescence at all. But then when the deviant stimulus appears, you see a large increase in fluorescence or an increased response to this stimulus. And if you're not quite seeing it, you can just kind of focus here with no activity, and then bam, when the deviant stimulus happens, you get this large response. So here is just a basic averaged response plot. Neurons are on the y-axis, and then time is on the x-axis. And these are just averaged calcium, first derivative of the fluorescence response to the onset of the visual stimuli. And as you can just see here, the average response to the redundant stimulus is pretty low, but to the deviant stimulus, you get a large response from perhaps more neurons and larger responses in the neurons which were active to the redundant to begin with. Anyway, so and Eric was getting at this a little before, and it's an important point to drive home, is that there's at least two things going on here. One is sort of what we call stimulus-specific adaptation. The neurons in V1 actually begin to reduce their responses to a repetitive stimulus, or a stimulus which is otherwise predictable in the current context. So you get this decrease in responses from the baseline level of response during the paradigm. But then when the unexpected stimulus comes in, you not only get a recovery from that adaptation, but you get a larger response than you would have to the initial presentation. So you kind of have this, you can imagine this bidirectional effect which happens during the oddball paradigm, and it's that augmented response which we're most interested in, because it suggests that the circuit is generating a prediction error or an added signal to a stimulus or an event it did not expect. So in order to sort these out, we use a relatively simple set of runs. Each one of these runs is about 12 to 15 minutes. We use a basic oddball paradigm, which I've already shown, with just like repetitive presentation of a particular orientation, and then the deviant stimulus is either the opposite orientation or a 45-degree rotated one. And then we use a flip-flop oddball paradigm. So in this case, the stimulus which was originally the deviant in the first run is now the redundant stimulus, and so this gives us the ability to focus on eliminating simple effects of orientation selectivity in our neurons, and then we use what is called the many standards frequency control, where we present a whole number of different oriented stimuli. And in this case, these oriented bars are neither redundant nor deviant in this context. There's nothing surprising about this orientation, but it is still rare. So by comparing cortical responses across these three contexts, we can measure responses in the same neurons to the same stimulus when it is kind of equally probable as other orientations versus when it is highly predictable or redundant versus when it is contextually deviant. And so that's what you see here across the horizontal. These are the same neurons across three different contexts. And you can actually see this bidirectional modulation just at the single cell level. You get a reduced response when that same stimulus is redundant in its current context, and then an augmented response when that stimulus is deviant in its current context. And here's an average across neurons from that data. And I'll show a lot of plots like this just as a basic quantification of this effect. So here is the average z-scored calcium imaging trace. The dark black line is the response to the stimulus during the many standards control. The red is the response to that stimulus when it's contextually deviant. And then the blue is the response to that stimulus when it's contextually redundant. And like I said, this sort of suppression that we see or we can call it repetition suppression, or we can call it stimulus-specific adaptation, involves a reduction in responses when that stimulus is redundant. And then the augmentation or the increase in responses, I'm going to refer to from now on as deviance detection. It's not in the sense that the mouse is detecting or aware that the stimulus is unexpected. It's just that at the neural level, we can say these neurons to some degree are responding to contextually rare stimuli more than they would have if that stimulus was rare but not unexpected. So just to summarize the beginning part, deviance detection is observable in basic visual cortical neurons, and it reflects a form of basic sensory prediction error. And I want to make that clear that it's sort of sensory or temporal prediction error, because prediction error is used in a lot of contexts and can take a lot of forms. For instance, well, I won't get too deep into that. But the thinking is that deviance detection at this level is a neuron level basis of the EEG mismatch negativity. So some work I did after this was to get a better understanding. Of course, that initial work was just focused on layer 2-3, but we sought to see whether or not these deviance detection responses were present across different layers of the cortical column. As you know, in V1, layer 4 is the main thalamocortical recipient layer, whereas layer 2-3 sends a large number of feed-forward projections to higher visual areas, as well as a lot of local recurrent circuitry, whereas layer 5 tends to send feedback projections to lower cortical and subcortical areas. So here in this study, we measured cortical responses across many different depths. In this particular study, we only really got down to about 530. In some cases, we got almost 600 microns in depth, but we did stop there. So again, just focusing on neurons in layer 2-3, and here I'm just showing pyramidal neurons. This is data collected from my lab at Georgia State. So we've collected quite a few cells, and at the population level, you can see that averaging over layer 2-3 neurons, we have a clear deviance detection effect. On average, you get a much larger response to a stimulus when it's contextually deviant, versus layer 4 and layer 5 neurons. While they did show adaptation to redundant stimuli, you see this kind of reduced response reflected in the blue trace. There was no augmented response to a contextually deviant stimulus. There may be something slight here in layer 4, but it's nowhere near as dramatic as the effect we see in layer 2-3. So Looking a little deeper, we wondered, are these deviance detection signals kind of observed in all layer 2-3 neurons which respond to that orientation, or does this reflect a subtype of neurons present? So this is a basic scatter plot here. Each dot is a single neuron. And on the x-axis, I've quantified stimulus-specific adaptation. So this is the degree of reduced responses to stimulus when it's contextually redundant. And on the y-axis is a computation of deviance detection. So this is how increased responses are when that stimulus is deviant. So this is response to deviant stimulus minus control. And so just kind of eyeballing it here, you can see that there may be a subcluster of cells which show much larger responses to a stimulus when it's contextually deviant. So here's just a plot of, if we take those two populations, the black cells and the red cells here, and we average over them in a single mouse, and we look and see what their responses are during the control run and the oddball run, you can see when I turn the display on here, both subgroups of neurons fire quite a lot. Then once the control run starts, you get some regular response in both populations. But these deviance detector neurons seem to be showing a dramatically increased response only to the deviant stimuli, whereas your non-deviance detector neurons, while they're still showing reliable responses during the control run and some responses during the oddball, they don't show an augmented response to the deviant stimulus. And these are kind of some single cell averages to back this up here. Here are these kind of, as you can see, that the non-deviance detectors, it's not they like move out of the field of view or anything. They're still active during the oddball run. They just don't seem to show any preference towards contextually deviant stimuli. So to kind of follow up and test this in a little bit more of a rigorous way, we ran the same oddball paradigm 30 minutes later in the same neurons. And you can see if you kind of selecting for deviance detectors in the first run and then seeing if they still retain the same preference later on. These are the averages for those neurons. As you can see, there's some slight decrease in responses to the redundant stimulus from its first presentation all the way through. And then when that stimulus is deviant, you get a much larger response. So this sort of cross-validation confirms that these neurons do have stable deviance detecting properties. And the non-deviance detectors do not, they don't like sort of develop deviance detection out of nowhere. They have stable non-deviance detecting properties. So, one possibility here is that I've drawn these clusters kind of in a hands-on way. So, what if we just kind of use k-means clustering in this space and see, well, maybe there are other subtypes of neurons that we've inadvertently averaged over one another. So there's more than just deviance detectors. There are redundancy detectors and the inverse. Well, so this is with a k-means clustering of k equals 3. So you can see the deviance detectors pop out here, and they're still stable. But using the same cross-validation step, recording the responses 30 minutes later, these other clusters don't really retain any sort of contextual preference. They're still showing reliable responses to that same visual stimulus. It's just that they don't seem to care whether it's redundant or deviant or neither. And so if we repeat this same k-means clustering analysis with k equals 2 through 6 and beyond, the deviance detecting subcluster sticks around every time, and it sort of stabilizes to this 10 to 11% number, suggesting that the kind of true deviance detectors subgroup perhaps maybe only make up about 10 to 11% of the responsive neurons in layer 2-3. Notably, these aren't of all pyramidal cells in layer 2-3. These are of the neurons which responded significantly to this particular visual stimulus in any context. But in none of these cases does another cluster with a different type of context preference emerge. So we sort of concluded that perhaps about 10 to 11% of visually responsive neurons in this paradigm show deviance detecting properties. If we then kind of zoom back out and look again at the layer 4 and layer 5 data, it appears there may be in layer 4 a few neurons which pass cross-validation, maybe about 4%, but it's pretty low. And it's possible that these that our layer definitions were not perfect. We based this mainly on depth. So it is pretty clear that these deviance detectors are a layer 2-3 phenomenon. Another thing to point out is that these you know, we can call these context preference or deviance preference. It doesn't you know, relate in a straightforward way to orientation preference. So it's not just another proxy for very strong orientation selectivity. Here on the x-axis, I've plotted orientation selectivity in a totally different run with multiple orientations. And on that y-axis is a quantitative measure of deviance detection as the sort of response to the deviant versus control. And what you can see, these red cells are the ones which pass a cross-validation test. So these are your stable deviance detectors. And as you can see that there are quite a few deviance detectors which show very strong orientation selectivity. There are also quite a few neurons which don't exhibit deviance detection that have strong orientation selectivity. And then likewise, there are plenty of deviance detecting neurons which don't show very strong orientation selectivity at all. What was true is that it was rare to find a deviance detecting neurons which exhibited deviance detection to both to orthogonal orientations. So there is you know, there is some relationship to orientation selectivity, but one does not purely predict the other. So one particular thing we want to look at moving forward are these kind of deviance detecting or prediction error neurons in layer 2-3 distinct in some molecular way or in terms of which genes they express. So what we like to do is sort of identify deviance detecting neurons online in awake head fixed mice, and that's what these set of plots here are. Identify them using some threshold, and then go potentially try to photo tag them and see if we can sort them and run in a single cell or bulk RNA sequencing to maybe see if they express a different subset of neurons. And sort of just looking at the publicly available Allen Institute RNA-seq database of cortical cell types, what's interesting is that in layer 2-3 neurons in V1, there do appear to be strong evidence of some subclusters among excitatory pyramidal cells. So one potential possibility is that some of these subclusters might actually map on quite well to deviance detection. Okay, so that's this first part, and so as we know, layer 2-3 in the visual cortical system tend to feed forward projections to higher visual areas. This is somewhat consistent with sort of Carl Friston and colleagues and a number of others predictive coding framework. And to put this briefly, it suggests that the brain continually generates sort of an internal model of the environment to more or less predict sensory information coming from the world. And cortical inputs from the environment are compared to these internal predictive models, and sensory information, which is fully predicted by the sort of internal models at higher levels, is suppressed in the cortex, whereas sensory information or sensory inputs which are not predicted by the kind of ongoing model feed forward and generate what are called prediction errors. And so the idea is that this is happening at multiple cortical levels all the time. And so this is at least somewhat consistent with what we see about in terms of our deviance detecting neurons residing mainly in layer 2-3. But another part of this is the feedback from higher areas. So we asked what potentially what could be the kind of activity dynamics of top-down inputs to visual areas during this paradigm. So we focused first, instead of on kind of LM or other higher levels of the visual cortical hierarchy, we went even further up and looked at an area in mouse prefrontal cortex termed the anterior cingulate area. This area actually projects direct inputs straight back to V1 and a number of other visual area, layer 1. And others have shown that this circuit is engaged during visual attention and during kind of reward prediction error and things like this. So we asked, what could this circuit be playing a role in kind of this more passive processing of sensory context? So we expressed GCAM in prefrontal areas, and then imaged the activity of these inputs to V1 at the axon or at the bouton level, and then quantified what these top-down inputs are doing during the paradigm. So here, you can see these are just some example traces here in C of what these axons are doing. And this data here is averaged over about 550 or so ROIs. We combined the different boutons or axonal segments that showed highly correlated activity, suggested they came from the same cell. So there were, in fact, way more ROIs than we analyzed. But as you can see just from this average plot here, while these top-down inputs showed very kind of reliable, stable, visually driven activity during both the control run and the oddball, they did not seem to show any sort of adaptation to redundant stimuli or augmentation to deviant stimuli. So PFC input does not appear to be modulated by context. It didn't show deviance detection, augmented response to deviant stimuli or stimulus-specific adaptation. This is this plot here. This is just taking all of the bouton ROIs and normalizing them relative to the response to control. And so here, the black line is the average response in V1 neurons to the redundant stimulus. So the dotted line there is their average response in the control case. As you can see, V1 neurons show reduced response. That's the stimulus-specific adaptation, whereas the PFC axons showed more or less the same magnitude of visually driven activity. But what's interesting is that they seem to start firing a good 100 milliseconds before the onset of the stimulus in this paradigm. And the same is true here for deviant, prior to deviant stimuli. If anything, it's slightly higher prior to the deviant stimulus, although it wasn't significant. But here again, in the black line is the average response of V1 pyramidal neurons. As you can see, it's larger than the response to control. But the PFC axons, the peak magnitude was about the same, but they are active prior to the onset of the stimulus. So this suggests to me that while they may not be, sending the deviant signal, and they may not be sending purely the directly suppressing the visually evoked response in V1 via like inverse redundant or stimulus-specific adaptation. They may be showing this kind of pre-stimulus modulatory role, which modulates the activity of V1s prior to the stimulus. So we further sought to understand the kind of temporal dynamics of this circuit. So in my lab, we recorded from bipolar electrodes, kind of locally referenced, approximately 300 microns between the lead and the reference, both in mouse area PFC and mouse area V1. And we sort of recorded the visually evoked activity as well as the ongoing activity during this paradigm. So this is sort of the average visually evoked potential to the moving grading stimulus in this paradigm. And here is just kind of the ongoing PFC response. We don't see an extremely large visually evoked response in PFC. However, we analyzed how the two synchronized during the paradigm. So we calculated phase coherence, or the consistency of the phase lag between oscillatory activity in PFC and oscillatory activity in V1. So here is a plot where time is on the x-axis and frequency is on the y-axis. And this heat map reflects how locked in phase or the consistency of the phase difference between V1 and PFC. And this was recorded, and this here shows is about 7 different mice in the paradigm. And there appears to be a highly consistent kind of shared signal or a phase locking between the two areas, particularly in this sort of theta to alpha range from about 6 to 10 hertz. And this is relatively consistent across the paradigm. So it's not necessarily made that much stronger by the onset of the visual stimulus. It's present prior to the stimulus as well. And here is just kind of an averaged over the pre-stimulus period. And you can see that the main peak here is at about 10 hertz during the oddball paradigm. And the phase lag was on average between the areas about 46 degrees or an eighth of a cycle at 8 hertz. So this could potentially be about a 15 millisecond lag in signal. I'm not quite that I don't think that's, I think it's a bit longer than it would take for the actual signal to go from one area to another. So I'm not fully sure on what that could mean, but it is it's relatively short. So in this sort of interim summary, I just want to say that PFC input to V1 during the oddball is kind of consistent or constant regardless of the context of the visual stimuli. It appears to be preparatory, so it's present prior to the onset of the stimulus. And it occurs primarily in the theta alpha band, and it's strongest there. So the next question is this top-down input to V1 necessary for deviance detection in V1? So in order to test this, we used Arch-T to disrupt or suppress prefrontal inputs to V1 during this paradigm. And so here, we use the viral strategy. We express Arch-T driven by the synapsin promoter in PFC, and then we did calcium imaging in V1. So this is recording neurons in V1 and suppressing inputs from PFC by shining an LED on layer 1 of V1. And the idea was to record V1 during the oddball paradigm when the circuit is unperturbed, and then again during a subsequent oddball paradigm during which half of the stimuli are disrupted. Okay. So here, what we saw here on the baseline, you can see V1 neurons showed, again, going to reduced responses when the stimulus was redundant. That's the blue trace, and augmented responses when that same stimulus was contextually deviant. However, when the LED was on, it appeared to disrupt deviance detection. On average, it did slightly increase responses to the redundant stimulus and the control stimulus, potentially by prolonging their activity, but it removed the deviant detection, or removed the augmented response to the unexpected stimulus. So when we suppress this feedback, we disrupted the deviance detection response. So PFC feedback is needed for distinguishing unexpected or novel stimuli in a basic oddball paradigm. And I haven't shown this, but I want to say that this effect was particularly prominent in our neurons which showed deviance detection. So to put all that together, deviance detection is potentially a form of sensory prediction error, and it could be a neural component of the mismatch negativity. It's primarily present in layer 2-3 neurons, and even subsets of them. Cortical feedback from higher prefrontal areas modulates V1 responses, and this is particularly strong in the theta alpha band. And it appears, this top-down input appears to be necessary for visual deviance detection in V1. So in this last part of my talk, I just want to present some data in which I'm starting to sort out how this is carried out. So this effect on V1 neurons appears to be somewhat bidirectional in the sense that suppressing PFC increases their responses during to the sort of non-deviant stimuli, especially during the control run, but it decreases their V1 neuron responses when that stimulus is deviant. So this implies that local circuitry may play a role. So this is where I'm going. They remember that, and so what you see here is somewhat of a complex looking little circuit diagram for how we think augmented responses are elicited during a basic visual oddball paradigm. Recall that feed forward inputs from layer 4, and I haven't shown it, but we did do some Bhutan imaging of LGN inputs to V1, and they also show a similar kind of adaptation to redundant stimuli. So you get reduced responses to reduced inputs to pyramidal neurons that prefer the redundant stimulus. But in this model, the thought is that unadapted pyramidal neurons which are selective for different orientations, appear to become hyper-excitable, such that when the deviant stimulus appears, they fire off a much larger response. What we think is that this may be due to increased suppression of somatostatin neurons, or release of inhibition from somatostatin interneurons. In that suppression of SSTs in this paradigm could be mediated by this top-down circuit, which may increase activity of VIP interneurons, indirectly further suppressing SSTs, which releases certain pyramidal neurons from inhibition, allowing the augmented deviance detection response. So that's a little bit complicated and multi-part, but the idea is that both adaptation in the feed forward inputs only to neurons which prefer the non-deviant stimulus, combined with some increased inhibition of somatostatin neurons from top-down sources, releases the non-adaptive pyramidal neurons so that they show the deviance detection response. So in order to test this, we started to record interneuronal activity during this paradigm. Here are pyramidal neurons in layer 2-3 of V1, and you can see this is the response to the control is a decrease during the redundance, and then when the deviant stimulus appears, you get the augmented response. I've shown you this already. What somatostatin interneurons do is they actually appear to show a much stronger adaptation to the redundant stimulus. And I'll show you another plot in a second that even emphasizes that. And they do show actually some deviance detection, but it's a little later than the pyramidal cells. And then what we see in VIP interneurons is actually relatively modest adaptation to redundant stimuli. And we don't actually see a deviance detection response. Now, note that the eLife paper was brought up previously from the Allen Institute study, suggesting that VIPs in that study did indeed show novelty detection responses. Notably, in this paradigm, the mice were not trained to do any task, so there was no behavioral relevance to these deviant stimuli. These mice weren't trained to perform a change detection task, and I think that really is what makes the difference here. Okay. So this is a plot again, just to show the same data, but relative to the pyramidal cells relative to their control response, which is the dotted line. The response to the redundant stimuli, you can see a much larger adaptation in the SSTs, and some late deviance detection in the SSTs, suggesting perhaps they inherit their deviance detection from local pyramidal cells. Whereas the VIPs didn't really show large modulation in either sense. And this fits with the model, miraculously, although the model I did come up with after the data, so I'm not gonna BS there. But what this suggests is that enhanced adaptation among somatostatin neurons may play a large role in this in disinhibiting pyramidal cells. And this may be due to the fact that VIPs are not adapted during the paradigm. And notably, neither were axons from PFC. So VIPs and PFC inputs showed basically the same lack of adaptation to the redundant stimuli, which fits in this model. So I just have a little more time, and I just want to say, which I think is the most exciting part of this. What we did is we sought to see whether driving PFC inputs to V1 should differentially activate these subtypes of cells. So here we imaged different interneurons, somatostatin neurons, VIPs, pyramidal cells, and we expressed excitatory autogenetics, channel rhodopsin, in PFC. So the idea was to stimulate PFC inputs to V1 at different frequencies, holding the kind of overall illumination power constant for brief bursts of 1 second, and then image neurons in V1. So we're driving PFC inputs to V1 and measuring V1 neurons of different subtypes. So here's just a movie of what it looks like. Because we turn the light on, we lose some data while the light's on, but because GCAMP6S is slow, we can make use of the signal after we turn the light off. And these are just some example of what V1 neurons are doing when we stimulate PFC. Of course, we're not stimulating these neurons, we're stimulating PFC inputs and seeing whether these inputs from PFC drive neurons differently. So this is what pyramidal cells do. And so here is when we, going from left to right, this is when we stimulate PFC with just block stimulation of weak or strong, and you get some degree of activity in pyramidal cells. But then once we start to get a little more rhythmic with 2 Hertz or 6 Hertz or 10 Hertz stimulation, we get a little bit larger of a response in pyramidal cells. And then once we step it up to beta range rhythms or gamma range rhythms, it actually starts to rebalance back out, and we don't get as strong of a response in pyramidal cells in V1. Now, when we record VIPs doing the same thing, we actually get not too much response at beta or delta ranges, 2 Hertz or 20 Hertz. We get a decent response in the 6 to 10 Hertz range, and then this very large response when we stimulate in the gamma range. And then when we look at the same thing in SSTs is where it gets really interesting, is that when we stimulate here in the 6 to 10 Hertz range and record SSTs, it actually suppresses SSTs, somatostatin neurons, but stimulates the other 2 subsets. I'm not going to get into what happens at 40 Hertz. I'm not quite sure how to make sense of that. But what's really interesting about this range is this is the peak frequency at which we naturalistically observed these two areas to be synchronous in. So when we drive at this more physiological frequency range, we suppress somatostatin neurons and activate VIPs. And again, this is just during spontaneous activity. And then notably, if we suppress VIP interneurons with chemical genetics, with DREADS, we actually selectively disrupt synchrony between PFC and VIP interneurons right in that range. So it leads us to believe that this kind of top-down input to V1 may engage VIPs, particularly at theta alpha bands. Let's see, I believe we have until 6:40. So I'm going to skip this overall summary and actually just go to the final slide here, where I thank everybody in my lab. I've been really fortunate so far at Georgia State to attract a lot of talent already. I have 4 PhD students and 1 F-32 funded postdoc, and everybody contributed to various parts of this. And I'm hoping to kind of keep digging further into this microcircuit to really understand, the basics of deviance detection and eventually mismatch negativity. So thank you.